Hello everybody, myself is SK Pal and I am from Shivaji Group of Institute. It is one of the most finest institute in the education system, provided best education. It also having a well educated staff. Myself is SK Pal. Today I am going to discuss a chapter of biology of class 12th and the chapter is molecular basis of inheritance. This chapter is very important for your board examination point of view because one or two questions are frequently asked in from this chapter. So in genetics, I told you that a molecule that is responsible for transfer the information from one generation to another generation is called DNA, deoxyribose nucleic acid. It is a type of nucleic acid called deoxyribose nucleic acid. This nucleic acid, that is DNA, is responsible for transfer the character from one generation to another generation. So in this chapter, molecular basis of inheritance, we will discuss all about the DNA molecule. How DNA molecule is, first of all, identify. A scientist called Frederick Mixture, first of all, describe DNA molecule. Frederick Mixture, first of all, describe DNA molecule. But how it can recognize that DNA is a genetic material, it is a question. So to solve this question, many scientists perform experiment which show that DNA is a genetic material. In 1928, a scientist called Friedrich Griffith Frederick Griffith first of all demonstrate that DNA is a genetic material. Frederick Griffith performed his experiment on a bacteria called Streptococcus pneumoniae. Streptococcus pneumoniae, it is also called, this bacteria is also called Diplococcus pneumoniae. It is also called Diplococcus pneumoniae. Frederick Mixture performed his experiment on a bacteria Streptococcus pneumoniae in 1928. It is one of the experiments which prove that DNA is a genetic material. Frederick Mixture take two strain of Diplococcus pneumoniae, The two strain is, first strain type 3 S strain, and second strain is type 2 R strain. Frederick Griffith take two strain of Diplococcus pneumoniae. First strain is called type 3 and S strain, and second one is known as type 2 R strain. Here S stand for smooth and here R stand for rough. Then what is smooth and what is rough? S strain of Diplococcus pneumoniae, S strain of Diplococcus pneumoniae is called smooth strain and R strain is called rough strain. In a smooth strain, what happened? This is a cell of Diplococcus pneumoniae. It is called smooth strain because it having a covering around it. And this cover of this bacteria is called capsule. It means that it is a capsulated bacteria. But in a type 2 R strain, the bacterial is made up of only in such a way that it does not having this capsulated covering around it. So what happened? This is called capsulated Diplococcus pneumoniae and this is called non-capsulated Diplococcus pneumoniae. It is called S strain, smooth strain, and it is called rough strain. This S strain of Diplococcus pneumoniae is virulent. And this strain of Diplococcus pneumoniae is non-virulent. Virulent means it having a nature to develop a disease. Develop a disease, but non-virulent Diplococcus pneumoniae does not develop disease. These two strains are taken by Griffith and perform his experiment. This experiment is performed in 4 steps. Here I am draw this diagram. This is very important diagram to understand the concept of Griffith experiment. This is called rough strain and this is called smooth strain. Rough strain is virulent in nature and smooth strain is smooth strain is virulent in nature and rough strain is non-virulent in nature. It means that the smooth strain provided disease in the mice. In the experiment of Griffith, he taken all the two strain, rough strain and smooth strain, and injected it into the mice. Because it take mice. Why it take mice? Because the anatomy and physiology of mice are very similar to the human beings. That is, it is one of the experimental material which is used in the laboratory to testing different types of medicines and different types of result. So rat are taken here. What happened? The rough strain, when injected into the mice by injected into the mice, and because it is non-virulent in nature, so the result will be obtained here. The mice is lives. It means that cannot take place of the mice. But when smooth strain of Diplococcus pneumoniae are taken and injected into the mice is dies. Because it is virulent in nature, so mice is died. Then After that, what happened when Griffith heat killed the smooth strand, when he heat killed the smooth strand, and this heat killed smooth strand are injected into the mice is live. And in his fourth experiment, what he can done, he taken rough strand, this is non-virulent strand, rough strand, and heat killed smooth strand. Heat killed smooth strand is non-virulent in nature, as you seen in this experiment, third experiment. And in fourth experiment, when he taken a mixture of rough strand and heat killed smooth strand and injected into the mice will die. Mice will die. So here what conclusions are coming? When the heat killed mice, when the heat killed smooth strand and rough strands are taken together, injected into the mice will die. So Griffith says that something is from the smooth strand of the Diplococcus pneumoniae passes into the rough strand, and this rough strand passes into the rough strand, and this rough strand show the nature of virulency in the mice. So the mice is die. But after performing such type of experiment by the Griffith, he does not find out what type of a molecule that is transferred from the heat killed smooth strand to the rough strand. He cannot find out it. After Griffith, three scientists, Avery, MacLeod and McCarty, proved that the substance that is responsible to the dies of a mice is a DNA, and it is transferred from one generation to the another generation of the mice. What is the result coming here? Heat killed smooth mice, when injected into mice, the mice will live. Why this happened? What is the reason that heat killed smooth strain is so non-virulence in nature, and rough strain plus heat killed smooth strain so virulence in nature? What happened? Actually, when the smooth strain of Diplococcus pneumoniae, it having a covering around it, and this covering is made up of polysaccharide, when heating should be take place, the polysaccharide covering of such a strain is destroyed, and it can be seen here. What happened? The heat killed smooth strain does not having the covering of polysaccharide. That's why it is non-virulent in nature. But what happened when the heat killed smooth strain is mixed with rough strain? Then this is rough strain and this is smooth strain. Something is passes from it does not having this heat killed smooth strain does not having the polysaccharide covering around it. Then what happened when these two things are mixed together? The DNA that is present in the smooth strain is passes into the rough strain, and the DNA passes into the rough strain, and this DNA is responsible to develop this DNA having a gene responsible to develop a capsule around it, capsule around it. Capsule around it. So this capsulated and it become capsulated bacteria. It become capsulated bacteria, and which is virulent in nature. Something is passes from heat kill smooth strain to the rough strain, and the rough strain become virulent. That's why the mice will die. With this experiment, it was proved that DNA is a genetic material, but Griffith does not explain it because he does not know about the DNA molecule. Ivory, McLeod, and McCarthy, these three scientists proved that the substance in the Griffith experiment was DNA. So, this is all over the Griffith experiment, and this Griffith experiment is called transformation. Transformation. So what is transformation? Transfer of DNA segment from the surrounding medium, donor medium, to the acceptor. So what is transformation? Transformation is a transfer of DNA segment from the surrounding medium, donor, to the acceptor. Thank you. This is all over the Griffith experiment.